Jai Hind everyone. Hope so you people are doing well. And this is the another lecture of a series of a DNA replication video. So this is the lecture 2B. So why I'm saying 2B? Because in the last video of the lecture 2, there because of some technical issue, we unable to complete it there. So we are continuing here again. So in elongation, we are going to see how the primer is working and what is the role of the DNA polymerase 1, 2, and 3. So we will discuss the eukaryotic DNA polymerase, the enzymes involved in the eukaryotic DNA polymerase, and then prokaryotic DNA polymerase. We'll talk about the DNA polymerase 1, 2, 3, 4, 5. We'll talk about the basic concept, and we'll talk about the primole, replizome, how the primer is put the new nucleotides, and the basic concept. So I'm starting from the beginning. I'm starting from the basic, that is the primer. So understand, this is a template strand. This is a template strand in which the new DNA is going to be synthesized. So this template strand have a two strand. One DNA having a two strand. One is the 3' to 5', and another is the 5' to 3'. Okay, another is the 5' to 3'. Now, if you see this priming, if you will understand, then it will be very easy to understand the questions. Sometimes they will ask the DNA polymerase have which type of prime polymerase activity. So it will be 5' to 3' polymerase or 3' to 5'. Why the lagging strand having Okazaki fragments? Why it is not a continuous? Why it is discontinuous? That is the concept we will understand. So this is a 5' to 3' to 5' template strand, and if you have to make a new DNA strand, then you need an enzyme which can be called as a, in different prokaryote it will be DNA polymerase 1 or 3, but in case of eukaryote it can be delta or epsilon. So epsilon will be on the leading strand. Let me show. Suppose this is a polymerase enzyme. This is what? This is a polymerase enzyme. Okay. Now this is containing what? DDP. Okay. Deoxy triphosphate. Okay. So DDP can be what? It can be ATP, it can be GTP, it can be ETP, it can be CTP. Okay. So anything it can be there, and it will incorporate this here. Okay. Now here it is a A. So what we will bring? We will take TDDP from here. Now whenever the new strand will come here. So this will be like this. Okay. It will have oxygen here. Then this is the 5', and this is the 3'. Now once you attach a new strand, this nucleotide having a free 3', which will be having, this will have OH group. Okay. So now this OH group is able to bind with any of the DDTP, and it will, once the new nucleotide will be attached with the condensation reaction, this one water molecule will be removed, and then because of this free OH group, it will be easy to form a new phosphodiester. Okay, but it is easy, then it will be easy to form a new phosphodiester. Now you can see there a new nucleotide is came with the A. Now this 3' will be again free. Again this 3' having what? OH group. If it is having a OH group, now a new nucleotide will again come, and it will easily bind with the, so like that it will again have a 3' which is having a OH. Now again one trinucleotide phosphate will come, and it will form so this, in such a way, this is the synthesis of a way of synthesizing a new nucleotide. So this is a way of synthesizing a new polynucleotide in which this alpha polymerase enzyme is involved in case of the eukaryote, but in case of the prokaryote it can be different. So this is alpha. It is an epsilon polymerase which is continuous in nature. What it is? It is continuous in nature. And now if you see, if it is a continuous in nature, then it is called as a leading strand. What it is called? It will be called as a leading strand. Where should I write? Leading strand. So leading strand is synthesized by what? It is synthesized by the epsilon. Now if you see another enzyme, so suppose this is a delta enzyme. How it is? It is a I'm just simply drawing a diagram. It's not exactly 2 delta. That's only one. So it is a delta enzyme. In this delta enzyme, what it is going to do? It is again going to do. It is again helping in the synthesis of a new strand. But we need a primer that we know. We will talk it later. But just I want to show you how this lagging strand is forming and what is the concept of the leading and the lagging strand. So this is the first polynucleotide. This is the first nucleotide here, the polynucleotide. Now this is the 5 prime. So what will become here? Here it will become 3 prime. Are you getting my point? So if you will start from here, the 3 prime will have. Let me draw. So now 3 prime is there. Okay. This is the 5 prime. So you know the polymerase activity will be always from the 5 prime to 3 prime. It is always from the 5 prime to 3 prime polymerase activity is there. Okay. Why? Because only 3 prime will have only OH group, 3 OH group. Okay. So if you added a A here, now once you will add a new nucleotide, is it possible to attach one more nucleotide here? Does it have OH group here? Think about it. Does it have a OH group? It doesn't have any OH group here, so it cannot bind a new nucleotide at this place. So what you will do? You have to start somewhere from here. Okay. So in this also, in this side there is no new template DNA, so it cannot synthesize here also. It cannot synthesize here also, so it have to start somewhere from here. So you given a primer, suppose, and this is a G. Now this is a 5 prime, and this is a 3 prime. Okay. Now this 3 prime have a OH group, and we have a template also. So we will attach one more nucleotide here. One more nucleotide we will attach here. Okay. Now again we have a OH group, so we can attach one more. We can attach one more, and like that we will have a gap. So this strand having a gap, that is called as a lagging strand. So like that we will have a small lag, and this is called as a Okazaki fragment. What it is called? Okazaki fragment. So this Okazaki fragment, or it is called as a lagging strand. This lagging strand is lagging because of lack of the OH group. Whenever the polymerase or new nucleotide will be synthesized, it will always synthesize from 5 prime to 3 prime. But in the template strand of 5 prime to 3 prime, it will have lagging strand. In the template strand from 3 prime to 5 prime, this is the template strand. Don't misunderstood with this new strand. Okay. So template strand 3 prime to 5 prime, it will have always 5 prime to 3 prime leading strand. But in the template strand 5 prime to 3 prime, it will new strand will having lagging strands, and this is from always 5 prime to 3 prime only. Are you getting my point? Okay. So this is the concept which I want to tell you, and you just remember this delta is for the delta polymerase, which is from the eukaryote. This delta polymerase is always synthesizing a new polynucleotide on the lagging strand, and epsilon polymerase will synthesize only on the leading. So gamma is helping in the polymerizing. What? It is helping in the polymerization of mitochondrial DNA. Okay, mitochondrial genome. So gamma is error prone. It is error prone. What do you mean by error prone? It doesn't have proofreading activity. And in this video only we will understand what is the proofreading activity. So delta enzyme you understood, and delta having a proofreading activity, and I will tell you what is the proofreading activity. Let me draw the prokaryotic enzymes. Prokaryote. In prokaryote, the DNA polymerase are of several types. What it is? 1, 2, 3, then 4, then 5, and so on. So these are the major DNA polymerase. So in beginning we were thinking that the DNA polymerase, only helping in the synthesizing a new strand, but just because of that Arthur Korenberg, who discovered the first polymerase which is the DNA polymerase 1. What it is? DNA polymerase. This DNA polymerase 1 having a 2 units. After the proteolysis, this DNA polymerase 1 is divided and break down into two parts. One is called as a clino fragment, another is the 5' to 3' exonuclease. A small fragment, another one is a small fragment which having a 5' to 3' exonuclease activity. Okay, exonuclease activity. And this clino fragment having the 3' to 5' exonuclease activity and 5' to 3' polymerase activity. Okay, what it is polymerizing? It is polymerizing a new DNA strand. So this is the 5' to 3' polymerase activity in the clino and the 3' to 5' exonuclease and 5' to 3' exonuclease. So why we needed two type of exonuclease? Let me know. Please tell me why we needed two type of polymerase. This is the DNA strand and suppose this is the 3' to 5' and this is the 5' to 3'. These are the template strand. Okay, these are the template strand. I am drawing this template strand in the blue color. Tell me on which strand this leading strand will come, on which template strand the lagging strand will come. So if your answer is that this is the 5' to 3', so in this leading strand will come and here the lagging strand. So this is again 5' to 3'. Okay. So whenever there is an immediate nucleotide adding which is damaged or which is wrong. So suppose you added a nucleotide here. This is a A, this is a G, and you added here instead of T, you added a C here and C. Okay. So immediately you added a C. Here it should be T. Adenine will bind with the thymine. If you don't remember, don't worry. But for now, you continue with me. So adenine will be binding with the T, and thymine is binding with the T, and cytosine is binding with the guanine. So this adenine is binding with the T, but here it is what? Cytosine. So this cytosine have to be removed, and this immediate nucleotide will be removed by the 3' to 5' exonuclease activity. It means it is checking the proof whether it is a correct or not. If it is a checking a proof, then it will remove this C and it will add T. Okay, and then it will add C here. So this is the way. This is the way by which it will be doing the proof reading activity. So this 3' to 5' exonuclease activity is what? It is a proof reading activity. It is a proof reading activity. Now, this 5' to 3' exonuclease activity, where it will be used and how it will be used and why it is used. So this 3' to 5' exonuclease activity is for the, tell me, this primer which is already added. These are the primer. What it is? These are the primer. So this primer have to be removed because it is made up of the RNA. Are you getting my point? Getting now? So this RNA have to be removed, and it will be removed just by the help of the 5' to 3' exonuclease activity. It is not able to remove the immediate nucleotide. Instead of, it will remove the prolonged nucleotide. Are you getting my point? Okay. Now this is called as a primer remover. Primer remover. Note it down here. None of eukaryote polymerase have 5' to 3' exonuclease activity. So you may be shocked why it doesn't have the 5' to 3' exonuclease activity. If it doesn't have the 5' to 3' exonuclease activity, then how they will remove this primer? That is a question. Must have to come in your mind, and you should ask me when you are taking a live class, or also you can ask in the comment box. So this is for you. So let me tell you that we will cover in the termination section. Okay, soon we will be able to cover this, how it is removed. But it is very important. You must note it down. None of the eukaryotic polymerase have the 5' to 3' exonuclease activity. Now, so you must have understand the DNA polymerase were discovered by the Arthur Coronball, and it having a 2 unit, one bigger unit and another smaller unit. And this DNA polymerase 1 have the proof reading activity 3' to 5' 5' to 3' exonuclease primer removal activity and 5' to 3' polymerase activity. Why this polymerase activity is there? It is for the gap filling. Okay, gap filling. So where is the gap? Where is the gap? Did you find any gap here? Of course, there are the gap. Once you remove this primer, there will be gap, and this gap have to be filled by the DNA polymerase 1. In case of the eukaryote, how it will be filled, we will see soon in this lecture only. Okay. Now I'm going to erase this, and let me know if you have any doubt. You can join our WhatsApp group also, where you can ask your doubt and you can send your question, as well as you can join our live classes and our app, where you can upload your doubt, and the questions which is asking here is very interactive. Now I have one question for you. Like, if there is a X protein, suppose there is a X protein, and you want to know either it is a exonuclease or endonuclease in nature, and you want to know whether it is cutting from the 3' or 5'. What are the experiment you will suggest, and what is the hypothesis you are going to give me to found that new protein you found, X protein, and it is an exonuclease or endonuclease, and second thing, it is cutting from the 3' or 5'. What are the experiment you are going to do? That is the one question. You have to give me the hypothesis or the experiment which is involved in this. Now, so DNA polymerase 1 you seen just now. DNA polymerase 1 you seen. This is for the, again, primer removal, and then what? Gap filling and primer removal, gap filling and proofreading. Now another is what? DNA polymerase 2. It doesn't have very much work, but it have a very important role during the DNA repair. So it will help in the DNA repair. So whenever the DNA repair is term come, so you have to think what it will repair. It will repair a damaged DNA. Damaged DNA means what? There will be unwanted nucleotide or mismatched nucleotides will be there, mismatched. So that will require 3' to 5' exonuclease activity, and 5' to 3' polymerase activity. Now the third one is that, and this third one is very important, which have a 3' to 5' exonuclease activity, 3' to 5' exonuclease activity. It doesn't have 5' to 3' exonuclease activity. Okay, and 5' to 3' polymerase activity. Okay, its speed will be around 1000 nucleotide per second. It will synthesize almost 1000 nucleotides per second, and you don't have to remember everything. You just remember which have what. So all the enzyme have the 5' to 3' polymerase activity. It's a common thing. No need to remember. Second thing is what? This 3' to 5' polymerase exonuclease activity will be having in all of this polymerase. Okay, it will be having in all of this polymerase enzyme. Now, this is the important thing, that 5' to 3' exonuclease activity is there only in the DNA polymerase 1, which you have to remember. Okay. So it is the main enzyme. This is the main enzyme, and it having following subunits. Actually, it having more than 3 subunits, but here we are discussing the alpha, beta, gamma. So the alpha subunit, I am indicating it with the circle. Okay, this alpha subunit is main polymerizing. Okay, polymerizing new strand. So if it is like it is a rope, there is a rope, invisible rope, and if you are there, okay, suppose this is an invisible rope, and this is our alpha polymerase. It need a balance. What it need a balance to walk over this template strand. For this needing a balance, we need what? A small stick-like structure. Okay, what we need? We need a small stick-like structure. Okay, so this is our beta, this is our alpha. We need something to hand over this beta that is provided by the okay, this is provided by the gamma. So I just simply give you the simplest illustration that alpha is the main polymerizing unit which is walking over the template strand, we need a balance by which we need a sliding clamp, that is a beta, and we need a slide clamp loader, which is a gamma. So beta is what? Beta is sliding clamp. Beta is sliding clamp holding polymerase strand. Okay. Another is the gamma. So gamma is for what? It is a beta clamp loader. It is a beta clamp loader. Beta clamp loader. So this beta clamp loader is loading the beta part, and also it will help in the attachment and detachment of the polymerase during on the lagging strand. Okay, attachment or detachment of polymerase on lagging strand. So this video maybe it is long, so please fast forward the video so you will have a better experience. Now we have to recover all these things, and you hope you understood this diagram. Will give you the table in the last of these videos, so you will be easily recall everything in just one table. Okay. So this is the common thing which I am going to tell you for the prokaryote and eukaryote. Okay. But initiation part, elongation part, the enzyme which is involved, that is I already separately told, and I am going to start with the initiation. Okay. So this is the initiation. We need a what? We need a helicase enzyme. Okay. We need what? We need a helicase enzyme to open this. We need a DNA A, DNA B. Or in case of another enzyme, we need RPA or MCM. Okay. In eukaryote, we need a MCM. So I am taking as a prokaryote. Okay. Prokaryote part. So we took a DNA B. Let me draw with the table type of diagram. That's not the table, and now this is the DNA B. What it is? This is the DNA B. Once it will be helping, it will helicase enzyme. Helicase enzyme. It is opening the DNA. Okay. So once it will be single stranded, the DNA is a single stranded, then it can be degraded by the nuclease activity. What it is? It can be easily degraded by the nuclease activity. So there we need some important enzymes which is called as SSB. What it is? Okay, this is the SSB protein. SSB protein. What is SSB? Sima bulk. Okay, it's not that. It is a single strand binding protein. Okay, it having an accessory protein. Replication protein. Replication protein. Okay, replication protein A. So this replication protein A is there, which is helping the single stranded binding protein. What it is doing? It is preventing from the nuclease activity. Okay, preventing from nuclease as well as coiling. So it is a coiling written here. Please note it down. So you made this SSB, and then you found RPA, replication protein A, which is involved in the mediating this single stranded binding protein. Okay. Now what is the next? The next is that polymerase enzyme will come. Okay. So what polymerase enzyme will come in beginning? And then like polymerase 3 is coming, suppose. Okay. So what it is coming? Polymerase 3, which having a alpha, beta, and gamma. Okay. So we have to remove this. We have to remove this. This single stranded binding protein will be removed. Okay. Okay, so there was attachment here, but now it is removing from it. Okay, so what is helping in this removal? It is helped by a, it is called as a RMP. What it is? RMP. What it is? Replication mediator protein. What it? Replication mediator protein. So this replication mediator protein is removing this single stranded binding protein, and then next the replication, the final replication is begun. That is this is a 3 prime to 5 prime. This is a 5 prime to 3 prime, and here you will get 5 prime to 3 prime. Here you will get 3 prime से 5 prime to 3 prime. Okay, and it is done by what? It is done by the this is our polymerase. It can be DNA polymerase C, or it can, in case of eukaryote, it can be, if it is a leading strand, then it will be what? Delta or epsilon. Then it will be epsilon, and it is in lagging strand. In case of eukaryote, it will be delta. Now this is all about the DNA replication elongation part. We need, just before that, we need a enzyme. Let me draw here only. So after that, we need an enzyme which is called as a primase. What it is? It is called as a primase. Okay. Primase is a, actually it is a RNA polymerase. Actually it is a RNA polymerase, and it's little different from the regular RNA polymerase which we studied in the transcription. So this RNA polymerase recruits a RNA. What it is recruiting? It is recruiting the RNA. Okay, it is a primer. It is recruiting the RNA. It is recruiting the RNA as a primer, and after it is recruiting the RNA primer, it will be elongated by the DNA polymerase 3. But in case of the eukaryote, this primase having the alpha subunit. What it having? It having a alpha subunit of the DNA polymerase. So if it having a alpha subunit of the DNA polymerase, which is a, suppose we seen the DNA polymerase is of different type in the eukaryote. We seen alpha, beta, gamma, delta, epsilon. So delta, epsilon we already studied. But if this is a, this is what? This is eukaryote DNA polymerase. Why it is required? It is helping the DNA primer, RNA DNA primer. Okay, RNA DNA primer. So together this will call, together this structure. Okay, together this structure will call, it's called as a replizome. So what is there in the replizome? Replizome having a primer, primase enzyme. This is the primase enzyme. Okay, and then alpha subunit which is in the eukaryotic, and this polymerase activity plus primase activity will called as a replizome. Are you getting my point? Now if you see one alone, the primase, it is a RNA polymerase. What it is? RNA polymerase which is helping in the formation of a primer, which is a RNA in nature. And another is what? Alpha or any other polymerase which is extending the primer till 20 nucleotide. It is extending the primer till 20 nucleotide. So after that this polymerase will come, and then it is going to synthesize the leading and lagging strand. If you have any doubt, you can ask me anytime. So before discussing the termination, we will talk about the primer removal. In case of the prokaryote, the primer removal will be easy because it have a enzyme DNA polymerase 1 which is able to exonucleate, which have a nuclease activity 5 prime to 3 prime exonuclease activity, and it can cut the primer easily. But this DNA polymerase 3 have to be removed. So once it will reach to the primer, the DNA polymerase 3 will be removed, and then DNA polymerase 1 will come, and it will polymerize, and it will cut this primer, and it having a gap filling activity because it having a 5 prime to 3 prime polymerase activity, so it will fill this gap, and then the phosphodiester bond will be desynthesized by the help of the DNA ligase. This is the way how the DNA polymerase will be this primer removal will be occur in the prokaryote. But in case of the eukaryote, let me draw the diagram. So in case of the eukaryote, what we have to do, we have to importantly we have to draw like 5 prime to 3 prime directly. This is the template DNA, and this is my primer, and this primer having what? This primer is made because of the primase activity and alpha polymerase. So this alpha polymerase lack 3 prime to 5 prime exonuclease. What is meant? It lack proof reading. So it doesn't have proof reading activity. So this alpha polymerase synthesize so much of the false DNA. Suppose this is a T, they added here G. Suppose this is a A, they added C. This is a C, they added here also A. So such type of mismatch repair have to be removed. This have to be repaired. So this alpha polymerase lack this proof reading activity. That's why it did wrong polymerization. Let me show how it will be rectified. So this this wrong mismatch, this mismatched DNA have to be removed. So in this case, the delta DNA, this is the Okazaki fragment. So this delta polymerase is synthesizing. Along with this delta polymerase, there is a helicase will involve in it. Okay. So now this helicase will come, and you will find it is like a crane which is removing this endonuclease. Okay. So which is removing this primer. This primer is removed. This is the helicase along with the delta polymerase. So this primer is removed. So this primer is removed, and now once this primer is removed, we cannot cut here because this delta have only 3 prime to 5 prime exonuclease, but doesn't have any 5 prime to 3 prime exonuclease. None of the eukaryote polymerase have the 5 prime to 3 prime exonuclease. So we need an enzyme which is called as a flap endonuclease. What it is? It is called as a flap endonuclease. Flap endonuclease 1 enzyme, which will cut this portion. After cutting of this portion, you will find a gap, and this gap will be synthesized by the DNA polymerase delta, and this delta polymerase again finally synthesized, and this gap will be filled. So this is not a gap, it is actually a need of phosphodiester bond, and this is done by the DNA ligase. So DNA ligase will come, and it will fill this by the phosphodiester. Okay. Now you will get a new DNA strand. After the removal, you will get a new DNA strand. Now we have to terminate this, and the termination will be done. The termination will be done only by the TUS protein. Okay. So we need a termination sequence in case of the prokaryote. If you will find a termination sequence, it will be easily incorporated the TUS protein. Where this TUS protein is so termination will be done by the TUS protein. This TUS protein having a 2 orientation as we shown. So if it will pass from one TUS this orientation, the fork will easily pass, but it cannot cross this from this portion. So in such a way, this 2 fork will come and meet at the center, and finally this new DNA strand will be formed. And it will be easily this interlinked DNA strand will be released by the topoisomerase 4, which is a type 2 topoisomerase. But in case of the eukaryote, there is no such type of termination sequence still known in every eukaryote. But in case of the yeast, we found there is a related termination sequences present in the yeast. Okay. Another thing is that there is a replication foci which is consists of the 10 origin of replication at least, and this 10 origin of replication will mediate the termination without the termination sequence. And once the termination will be done, the topoisomerase 2, that is a type 2 topoisomerase, will release this interlinked DNA in case of the eukaryote. So this is all about the DNA replication. This video lecture 2 is also finished, and in the next video we will bring the telomere replication and then research article discussion, and then we will discuss the question paper. Okay. So question paper and DNA replication related question paper from the Indian examination. Okay. And then there are several examinations for the fellowships for that GATE or CSIR or the ICMR or other examination we will see, and we will see other questions from the different parts of the world, and we will try to solve that, and we'll discuss that question in the video. Thanks for watching. We'll meet in another video. Stay tuned with me. If you like this video, please do share and subscribe.